use his idea to use about 20 minutes to finish the slides, and give more time for us to discuss. So this is very simple. We're trying to find out a new biomarker for the oxygenic photosynthesis. So this is the general history about the rise of the atmospheric oxygen. You can see that the rising of atmospheric oxygen is a very important process in the history of life of Earth and the history of geochemistry in the whole on Earth. Here is about the biological evolution at early time. There are some bars going to the very early time. We actually don't know what's exactly was the time with the origin of cyanobacteria and the other microorganisms, especially those cyanobacteria, because there are many mineralogical and biomarker evidence of them, which are very controversial. And here is people using the biological clock to calculate the emergence of cyanobacteria. Let's say it's somewhere around before the Great Oxygen Event, and about 3 billion years. The atmospheric oxygen is very important for the evolution of the whole biosphere, because this is the profile of UV radiation. You can see that the UV radiation is before the emergence of the ozonosphere. All the UV radiations, including UVC, UVB, and UVE, could reach to the surface of the Earth that can directly kill all the microorganisms on the surface of the Earth. Like this. This is this UVC, the short wavelength, the UV radiation, UVB, and the UVA. Now only UVA can reach to the surface of the Earth. UVA is very deathful. It can destroy all DNA and can kill microorganisms. So this is just to show you that actually the UVE kill all the cells on the surface of the animal. Here, this is the global distribution of the penetration of UVB to the water. You can see very clearly that the microorganisms at the photic zone can actually very effectively filter UV radiation, because you can see that here is this is upwelling zone offshore Peru. You can see that the plumes going this way. That with the high density of microorganisms, the penetration depth of UVB is very shallow when compared to the open ocean. So that this is the previous publication showing that the UV radiation. Now this is profile of present. So previously all those UV radiations could directly reach to the surface of the Earth. So that means that without the ozonosphere, the surface of the Earth, the terrestrial environment, and the near surface of the photic zone of the ocean could not be habitable for life, or those microorganisms. This is more calculations on the UV stress of the Archean life. You can see from here that after the formation of the ozonosphere, the primary productivity of the whole whole ecosystem can be increased dramatically after this time. This is some other results showing you that even with the formation of the organic haze, it still cannot be an effective way to filter short wavelengths, the UV radiation, to the surface of the Earth. As we are looking for the biomarkers for the oxygen-producing photosynthesis. So we know that before that there are already some other macroorganisms. We put them all in the category of chemotrophs because they use geochemical energy and carbon source. So that now we believe that, especially with the recent publication, that those chemotrophs, they should be earlier than photosynthesis, than oxygen-producing photosynthesis, even those photosynthesis, because the photosynthesis, they use high technology to harvest those photon as the energy source. So this is the comparison of the using the photon as energy source, and just use the geochemical energy as the energy source. You can see it's very different. So when this is use the photon as an energy source, so when with the depth of the ocean, that the biomass decreased dramatically because of the lacking of the energy source for those microorganisms. But if the microorganisms use photon as the energy source, there will be a lot, and the energy will be every day for the Earth. So we also we prefer to look also the biomarker directly by using electron microscopes to do the direct observation. So we checked the oldest sedimentary rocks, and we also do some other some mineralogical studies, and we have recent paper showing you that in the oldest beef that those iron oxides, especially magnetite, could be the result of a interaction between the iron 2 and the ferrihydrite, which could be the result of photosynthetic. And this is we do the detailed petrological study. We suggest that the oldest beef is a lithological evidence of life, of the earliest life. And I think this is something related to the previous talk, that this is our observation of the graphite particles. This is around a few micrometers, and it's coexisting with phosphate, with amorphous phosphate. So this is probably something Stephen Moses suggested, that if there is no biomarker, we can check out the mineralogical assemblage of carbon and phosphorus. And so this is clear evidence of the earliest photosynthesis. It's stromatolite. But there are recent studies questioned this as the evidence of the oxygen-producing photosynthesis, but it still could be the evidence of the anaerobic photosynthesis. Okay, and this is previous studies using biomarker as evidence of the oxygen-producing photosynthesis, but it's not reliable after the examination. And so also there are some biochemical studies found that all those biomarkers could be produced by the anaerobic photosynthesis, so they are not the actual biomarker or oxygen-producing photosynthesis. But there are also some other observations that are still showing you that the oxygen-producing photosynthesis could still be produced, could be emerged before the last GOE, Great Oxidation Event. So this is a model of the different types of way that can produce precipitated iron 3 minerals in the ocean. One is anaerobic photosynthesis, another is oxygen-producing photosynthesis. The difference is one uses oxygen, one uses enzyme. This is, by the way, we observed those banded iron formation with high-resolution SEM for those minerals. you can see the difference between the through time. This is latest from the this is lower Cambrian sedimentary rocks. You can see that organic matter is very clear with clear signals of organic carbon. Here is just it's around 2.5 billion years. You can see a few signal of carbon, but for those 3.7 billion years, you cannot see any signal of carbon. It's all mineralized to oxides. Okay, but we still can directly observe the organic matters in the banded iron formation, especially on the surface of magnetite. You can see this is a clear piece of organic matter. And we also did some synchronous radiation to measure the valence states of manganese. We found some of them can be very interesting. That in those on the structure of magnetite and the structure of hematite, we still can see we can find some manganese 3+ and manganese 4+, but we still don't know if this could be the direct oxidation by oxygen or by enzyme. And this is the observation of the signals of organic carbon in the magnetite from banded iron formation. So this is the spectra are very similar to this magnetite produced in the lab by using the ion-reducing bacteria. Now, this is the molecule we recently studied. So this is the cytalamin, which is a UVR-resistant pigment. So it's so this is the pigment not used by the microorganism to harvest photon, but they use this to filter UVC and UVB before they do the photosynthesis. The molecule is very robust. It can be it has a melting point of above 325 degrees C, so that probably it can survive the relatively high temperature of metamorphism. And at this time, and at this temperature, all the other organic matters will be gone. So that we thought that this could be a potential biomarker. So this is the structure of cytalamin. So we do directly observation and also use some Raman and FTIR for the measurement. And finally, we didn't find any good signal to confirm the existence of this cytalamin in the banded iron formation of all the age, like from 2 to 3.7. But we do some calculation of the structure of this cytalamin. That we found that it's probably because the decomposition of cytalamin can produce all the derivatives. All the derivatives, they have very different profiles when compared to the profile of cytalamin. So that's we think that's probably the reason. But we also suggest that here you can see that the profile of those cytalamin and its derivatives was similar to the profile of kerogen. So we turn to another question, that if those pigments could be the precursor of kerogen, because all the other organic matters could be very easily decomposed completely in the sedimentary environments. And this is another interesting result we get. This is the structure of cytalamin, some derivatives. So this is reduced, and this is oxidized. So when we do the calculation of the absorption of the cytalamin of UVC and the UVB and the UVA, it's very interesting that all those it and its derivatives, they can absorb UVC very effectively. So we suggest that probably this is with this result that microorganisms at a very early time, the photosynthesis at a very early time, they cannot reach to the very surface of the ocean, but with that pigment, so that the pigment can effectively reduce the UVC, so that they can gradually get to the surface, the photic zone of the ocean, and eventually to the surface of the terrestrial environment at a very early time, without having the effective ozonosphere. Okay. So that we think that with the production of that pigment, cytotonin, the microorganisms, the habits can be extended to the up photic zone of the ocean and even the terrestrial environment. And those phytoplanktons, they could block UVB that can further enhance the marine productivity. And the terrestrial mating habits that can enhance the making of the biofilms on the surface of the terrestrial environments and making the soils. This is a recent publication. It's about the observation of the soil, probably soil at about 3 billion years. So that if the microorganisms can live on the very surface of the terrestrial environment at about 3 billion years, that means that they also have the strategy to deal with UVC and the UVB radiation, because 3 billion years ago is a time that the ozonosphere was still not formed. So that this is our conclusion, that we think that before the formation of the ozone layer in the Archean, UVR, this is a fact, can reach to the surface, to the Earth. And just the previous view was that this life could not live on the terrestrial environment and the very near surface of the ocean. So our calculation shows that this pigment can effectively filter UVC and probably UVB. And so that means that a microorganism can live on the very near surface of the photic zone of the ocean and even the surface of the terrestrial environment without the protection of the ozonosphere. Okay. And with that, if this is correct, that the Archean primary productivity could be very high when compared to the our previous thought. Okay. That's it. Thank you.